I would like to draw a parallel about how we do things, and then how you look at it from the chemistry angle. So I'm sure, as academic institutes, you've heard about a lot of things that the government is doing. And to be honest, about 15 to 20 years ago, when we looked at the elite academic institutes like, ACTRIC or TIFR, they used to say that we are into basic research. So basic research, for example, in Cancer Research Institute, they would do mechanism of action studies or pathway studies. And all these, I would say 90% of such projects are funded by the government, DBT, DST, and so on and so forth. So what is happening is it's actually taxpayer money which is going into such academic projects. And I think at around 10 to 12 years ago, there happened to be a shift because the government realized that, a lot of taxpayer money is going into such projects, but there is nothing coming out. Like, for example, in a country like India, there are a lot of unmet needs. There are a lot of society problems which need to be addressed by a lot of research, which is happening in academic institutes. So the government came about with BIRAC. BIRAC is nothing but Biotechnology Industry Research Assistance Council. And as a biotechnologist myself, I feel that a lot of answers and solutions and keys to a lot of problems that we face today, whether it be in healthcare, environmental science, or agriculture, biotechnology holds the key to a lot of questions and problems that we are facing today. Now, academic institutes like yourself, you already have a mandate to carry out research. So say, for example, you might be having a project that you have to complete in order to get your degree, or you might have to do an industry internship. What's happening is this is sort of a bridge. So you come out of your academic lab and you get a feel and experience of how the real world works. Now, another advantage of academic research is it's more pure. For example, in industry, there may be a sort of a need to go about it for a commercial goal. How are you going to make money? Or they may say, okay, this may be a real need, but we are not going to make much money out of it, so it may be shelved. Having said that, from academia, it's more pure. The research is more pure, and you have the freedom to work on problems with greater risk because it's a safer environment. I mean, as students, you're not really, financially having any dependence. Teachers have, and lecturers, they have their salaries. So the risk is lesser. Consequently, you can work on a problem with a greater risk. You can take a greater risk. So PACE is nothing but, promoting academic research conversion to enterprise. So this is specifically, this particular proposal is specifically for academic institutes, and BIRAC has introduced this. You just have to do a proof of principle. So consider if you are in a 4-year course, by the first and second year, you decide that there are a group of students who would like to work on a particular problem. You have the guidance and wisdom of your professors and lecturers who have a wealth of experience behind them. So there is a lot that you can achieve. So you can easily do a proof of principle. You get up to 50 lakhs grant. Now, the advantage of this is you are actually de-risking the entire risk of starting a startup. So suppose you have an idea and you want to work with it, the government is actually giving you money to fail. So you can fail at the cost of the government. You have to look at it that way. You're not putting or sinking your own funds into trying out some idea. So This is one of the best benefits that I can think of. The second thing is it's better to be in collaboration with industry. If you have an industry partner who you can collaborate with, they can either give you funds for doing certain experiments, you can get into an agreement with them for, later on out licensing or sharing of profit or a royalty payout. And the IPR can be held either by the academic institute or it can be shared with the industry partner. The second stage that happens is the BIRAC BIG. BIG is the Biotechnology Ignition Grant. We got this funding in 2015. And this is again a grant in aid. That means the government is willing to give you the money, no strings attached. It's a grant. They don't want equity from you. They don't want anything from you. You own your IP. Now, where academicians come in is, say, for example, you are a professor or a lecturer applying for BIRAC BIG as an individual. The only thing is you have to have an NOC from the head of the institute that says that, you can take your idea and form it into an own company. You have to do it within the duration of 18 months. Again, the quantum is 50 lakhs, which is very generous to carry out your proof of concept. There is a catch. It is extremely competitive. Now, for all this funding that you're going to get, there are a lot of people out there with a lot of fantastic ideas. So at the time that we got the big funding, the success rate was 2%. That means if there were about 100 companies which, applied for the funding, only 2 companies sailed through. Now it has opened up a bit. I think the success rate is about 5 to 7%, simply because there aren't enough biotech companies out there. So what has happened is it's become a little more agnostic. It need not be purely biotech ideas. It can be chemistry. It can be any scientific idea. It can be a medical device. You know that in the COVID times, we have come across a lot of fantastic ideas which have come out there, right from telemedicine to monitoring, respiratory problems, non-invasive assisted respiration, and so on and so forth. So how you apply for this is to have your preliminary data ready. The preliminary data you would have generated hopefully through PACE or through an industry collaboration. And this is very important. Now, I'm not saying that I'm a reviewer of BIRAC projects. I'm not saying that just an idea will not get you. There have been people who have got funding for an idea, but then there are people who really have got a preliminary data to show that, yes, this is something which is feasible. This is something which is viable. So to compete against them without data is going to be very tough. The second thing is that anything that you are developing has to have a commercialization potential. That means if you are developing a product or a service, are there people ready to pay money for it? That is your commercialization potential. That also includes what is the market size? What is the market that you're going to be servicing or addressing? Another very important evaluation criteria is the pain point. So is there a gap that you are addressing? And these are all India specific, because we are talking about being in India. There are a lot of gaps, whether it's in healthcare or agriculture, marine biotech. So what is the pain point? What is the gap that you're addressing? And the most important is, can it be IP protected? The minute you know that you have something patentable, then you know you are onto a winning idea. So here I would like to use a bit of friendly advice. When you are doing your research, don't be in a hurry to publish. Go and see if there are others who are doing similar kind of work. Have they come out with a similar kind of solution? Or are you doing something unique? Speak to your IP cell. I'm sure you have access to it. It's easy to get an IP attorney who is worth his salt, do a little literature survey and find out, can it be patentable? Do you have freedom to operate? Which are the territories that you are going to protect yourself in? If you have even a preliminary patent filing, the chances of getting your BIRAC big project are greatly strengthened. So this sort of begs the question, why academic institutes? I mean, the answer is there in front of you. Youth is the biggest resource you have, the highest energy of your life. This is the time that you can take a risk. This is the time when you're brimming with ideas. You are already learning about what is happening in the world in real time. It's not outdated. And of course, the government wants to create jobs. So around in 2000, when the government introduced this new subject, biotechnology, in degree colleges. So you had BSc Biotech, MSc Biotech, and then the government realized that there were children coming out of these colleges with these degrees and there were no biotech jobs available. I would say 95% of them either were stuck in a sales and marketing job or they went abroad, and barely about 2 to 5% got jobs in actual research. So the government wants to generate jobs. The government cannot do everything. So the best way to do is to encourage a startup ecosystem where these innovations come out, they get converted into a company and they actually generate jobs. And finally, the sad truth is a lot of inventions get stuck in academia and do not reach the natural conclusion. So this, I'm speaking from my personal bitter experience. My PhD was actually patented and it's still stuck in that academic institute. So what is the natural conclusion? You do not patent something or file a patent to say that, yes, I have a patent. No, the natural conclusion is it comes out of the lab and it actually meets an actual need in society. It could be a product, it could be a service, but that's the natural conclusion of any patent. So how did we do this? I would like to demystify how do you convert a prototype into a startup. So a little bit about us. We were incorporated in 2013 and we developed cosmetic and clinical applications of stem cells. That's an interesting story which I'm going to share with you. And in brief, our vision and mission is that we want to be globally recognized. We want to meet unmet needs, but we are in the arena of stem cells. So that is how we are going to do it. We're going to do it in an ethical manner and in a sustainable manner. Plus, we're going to adhere to the highest levels of quality and safety in manufacturing. So we ensure that the best product reaches our target customers. So the first thing that I would say is leverage your strength. What is your strength? That you have to identify. So in our case, like Dr. Shivani said, I am basically a cell culturist and a stem cell biologist. I've been trained at very elite institutes at the national level for stem cell culture. So you can give me any kind of cell, I would be able to grow it. And plus I have an experience in cell culture, I have an experience in stem cell biology, and I have experience in skin research. Now, Mathen, my co-founder, he takes care of the business side of things. He is a chartered accountant. Wilfred has been with me almost from day one, and he is now the head of quality control and quality assurance. This is our core team. So when you look at the strength of this team, then it's a natural progression that we would be looking at wound healing. So just to give you a brief about the different applications of stem cells, right from baldness to wound healing to rheumatoid arthritis and diabetes, it holds the key. We concentrated on baldness and wound healing, and that's what I'm going to share with you. So First, you need to do a little bit of background work. What is the gap that you are identifying? Is there a need? And again, is that going to be convertible into commerce? So this is what we have to identify. So when I was looking at wound healing, we looked at the statistic for burns and non-healing ulcers in India. In India, it's really dismal. I mean, 80 to 85% of victims are women, and still today they are victims of domestic violence. And of course, for diabetes, we have fantastic numbers where, we are leading the world in diabetic numbers. So we have ulcers. We looked not only at India, but we looked at global needs as well. Is there a need for advanced wound care? And the simple answer is yes. The second question that we looked at is, how is wound care happening today? How is advanced wound care? So it's a surgical process. You have insufficient skin donations. And again, skin donation is from cadavers. So we have had that experience because we did a collaboration with Masina Hospital in Mumbai. It's got one of the best burns facilities in the country. And we helped them set up a skin bank. We operated it for 2 years. And then last year, we finally handed it over to them for independent operations. So the thing is, even though you use cadaver skin, it's not actually going to regenerate your skin. And the second thing is, when you use autologous or auto skin grafts from the same patient, it creates a secondary wound site. So you have your primary wound site, and then you're creating another wound, which is adding to the trauma of the patient. And plus, the final thing is, this has a very poor functionality. So say your hand gets burnt and then it is grafted over, it's very weak, it cannot function, plus it looks really suboptimal. So this was what we were looking at. So this is what you have to do. You have to do your background research. If you can't, I mean, half the battle won is when you do your research. So for example, if you are a group of 5 students working on a project, it's so easy for you to reach, each one of you to reach out to 20 people, and you figure out who is your target customer. Is it a retail consumer? Is it a doctor or a surgeon? Is it a hospital? So you have to do that research. So 5 of you can get input from 100. It's a simple market survey that you do. And you find out, if I develop such and such product or a service, would you be ready to pay money for it? And that's your first answer. If it's yes, then you know you're onto something worthwhile. So this was what we realized, that conventional skin grafting has all these downsides. And then we realized that if we can develop our stem cell technology for deep wounds, these are, I'm not talking about superficial wounds, I'm talking about 2nd and 3rd degree wounds, or really deep non-healing ulcers, which is as good as a 3rd degree wound. The advantage is, you don't need an operation theater. For conventional skin grafting, the doctors have to wait 4 to 6 weeks for granulation tissue formation. We save on that time. You can inject the stem cells in a simple OPD procedure. You don't need donor skin. So you reduce the trauma of the patient. You reduce the length of hospitalization. You get a faster and more aesthetic healing. And because the treatment time is reduced, it is affordable. So we started out with looking at keratinocyte cultures, which are the epidermal stem cells. And that was our first, I would not say mistake, but it is a process of evolution. And that's when we realized that, talking to clinicians, burn clinicians, plastic surgeons, we realized that where they need help is in dermal wound healing, not epidermal. So if you look at deep wounds, once the dermis regenerates, then you can probably avoid fibrosis, which is the natural wound healing which takes place in adults. And you can actually get into real skin regeneration. So you have to adapt, you have to be flexible. If something does not work, then you have to go on to do something different. And that's when we came about with how do we do an actual stem cell based therapeutic application for advanced wound care. And what we did was we switched to using mesenchymal stem cells from the umbilical cord. So this was the study that we did in collaboration with Hafkin Institute and IMEC, which is a Belgian company. They did hyperspectral imaging for us. And this study has recently been published about the wound healing potential of stem cells, that is mesenchymal stem cells. And conditioned media is nothing but the proteins which we harvest from these stem cells. Now, there were three important findings that happened because of this study. The first one was we were actually injecting human cells in a rat model. And what we realized was there was no cross reaction or immune rejection in this study. In fact, the treated animals, they gained weight and they were healthier. The second finding that we found was the stem cells. If you look at the middle panel and the bottom panel, the stem cells were almost as good as the stem cell protein treatment group. So now, we were getting insights because we could circumvent. There are a lot of logistical and storage problems associated with using cells. So say, for example, when you store stem cells, you have to do it in liquid nitrogen, which is at -196. And then that has to be stored in a hospital. You have to train the doctors how to thaw it and inject it into the patient, right? Versus if you use just the harvested proteins, this can be easily stored at -20, can be thawed and injected immediately. It gives almost a similar efficacy as the stem cells. And also, you are mitigating the risk associated with injecting living cells into another human being. The third and quite an important and interesting finding for us from this study was that when the regenerated skin was observed by hyperspectral imaging, we found that the skin appendages were also regenerated. That means we suddenly were seeing hair follicles, we were seeing sebaceous glands. Now, if any of you have had a wound, you would realize that you have scar tissue. It will be bald. There will be no hair follicles there. There will be no sebaceous glands there. So suddenly we realize that we are sitting with a new insight which can give us another spin-off. So that brings me to another vertical, which was stem cell based cosmetic applications. So this was the next idea that we jumped onto. So we did a proof of concept study. This has also been recently published in Journal of Cosmetic Dermatology. We went one step ahead. We stabilized the proteins into a topical formulation. So instead of injecting the proteins, we now had a shelf-stable formulation which can be applied at home. And that sort of expanded our market to global levels. So to conclude, this was our journey. So I told you how I started. I identified my strengths. I took an idea, I ran with it. Along the way, we evolved. We got funding support from the government that helped us de-risk a lot. We also went through the evolution of our own product. We made a wrong turn. We did a self-correction. We came back. We developed something further. In 2018, we also got follow-up funding from BIRAC, that is a seed funding where DBT has actually taken equity stake in our company. And this again is a very, very small percentage. It's the cream of the big grantees which qualify for this. And then we have filed a variety of patents to protect our IP. And then this is finally what we ended up with. So we actually developed a stem cell platform technology, and we have certain products which are ready for market. There are others which are under development, either alone or in collaboration with our partners. So you see how we have leveraged ourselves and developed ourselves into a really strong IP-backed company. So I hope I have demystified a lot of how you convert a prototype into a startup. And I would like to encourage you to chase your dreams, because you're young, you can take the risk, and the country needs you, of course. So with that, I come to the end of my talk. And this talk has helped me. Like I said, it helped. We are always so neck deep in our research and, firefighting all the time as a startup, trying to figure out how to meet the next challenge. And when I get an opportunity to do a talk like this, I stop for a moment, I take a look back, and then we realize how far we've come. So thank you for this opportunity. And I kept this short so that we have more time for an interactive session. So I'm going to stop sharing.
Thank you very much, ma'am. It's truly insightful talk, and the story of therapeutics, right from developing the prototype to the product.
Thank you.
So, there are any questions from participants? Okay. Ma'am, we have some questions from ourselves.
Sure.
So, ma'am, while we are submitting any project to the Big Biotech grant, at what stage it should be?
So it depends on which proposal. If it is space, then of course you have to, in any project that you submit, you have to have at least little experimental data, why you're doing it, what is the novelty, going forward, can it get IP protection? So depending on, so PACE is more preliminary, BIRAC is the next, then it goes on to further CIBRI and BIPP and so on and so forth. As you are graduating to the TRL level, technology development level, technology readiness level. So it depends on that. So if it is PACE, then it can be preliminary. PACE is tough to get, but I'm sure you can do it. Also, another way of getting funding is through getting an industry sponsorship. So if you have a company which is having domain in that or a presence in that arena, then it makes sense to have an industry partner.
Thank you, ma'am. And another question regarding that stem cell research. So, what facilities or conditions that required to establish the stem cell research in academic institutes?
You need a basic cell culture room. So you would need a biosafety cabinet, inverted microscope, liquid nitrogen cryo cans, -80 for freezing, -20 for storage, a fridge. It's a basic cell culture setup. I can help you if you are interested. I can help you with that.
Sure. Thank you. So, Shivani ma'am, is there any question from your end?
No, sir. Actually, ma'am, sir has already asked you, and you responded that how important is the company is an industry collaboration for this BIRAC project. So, is that company collaboration useful during the filing of the application, or I mean, at which stage it has a major role to play?
No, it's not necessary at all. It's not necessary. You can do it independently. You can hold the IP independently. The advantage is, and the money also comes only to the academic institute. It will not go to industry. The only advantage of having an industry collaboration is, suppose you don't get PACE, then you have an industry partner to, fund your experiments.
Yeah.
The second thing is advantage of having an industry partner is you have a backup. So suppose your experiments work and you prove your hypothesis, you have somebody to take it forward. So that's the advantage of having an industry partner.
Okay.
Yeah, so sir, can I ask one question? This is Dr. Athole.
Yes, ma'am, please.
Sure.
Yeah. Good morning, madam and sir. It's a very good talk. Madam, I have few questions related to dermal wound healing. You said you require a healing at a dermal level, but at a dermal level, we have all the network, right? Vascular network. So exactly what you mean by, and which studies have you carried out to prove this?
Okay, so this has become a very scientific discussion. So basically, it stems from when you have deep burns which go below the dermis, reaching almost the subcutaneous fat, where basically what happens is the dermal fibroblasts are damaged. And what happens in fibrosis is this wound is a breach of the skin, right?
Yes.
So the body is in a hurry to cover that gap because it has to protect it from the external environment. So it is a very unregulated kind of healing that is happening. It may not have functionality. It will take a lot of time. So what we found out during our research was now dermal fibroblasts, there are disadvantages of working in it because first of all, the source is a problem. The second thing is that it may get rejected if you put it onto an unrelated donor. So when we started working with mesenchymal stem cells, we get it from the umbilical cord. So the advantage is it is non-invasive biological waste actually, which we are getting. We do it in an ethical manner using ethics committee approval and informed consent. It's a large, huge, lengthy process that we go through. And finally, when you grow these cells, they look under the microscope very, very similar to dermal fibroblasts. And they share a lot of properties with dermal fibroblasts. And this is why we thought that these cells could actually help in dermal regeneration. And that was actually proved by the rat study that we did. Plus, we've done a lot of, when you talk about stem cell research, it's a very vast topic. So you have to prove in as many ways as possible that this is going to go and do the work that it's supposed to. So that hypothesis was what we have proved. We have done potency assays, complete characterizations. That was our BIRAC study. We did a complete characterization and we showed that we can establish a mesenchymal stem cell bank, which can actually function as an alternative skin bank. That was our project.
Okay, yes, ma'am.
So I hope I have answered.
Yeah, madam.
Part of your question.
Yeah, this wound, they differ from the chronic diabetic wound, I suppose, right?
Yes. So burns is an acute injury, and non-healing also would come under chronic.
Yes.
What I have found is that our product meets the need of both acute and chronic.
Chronic wound. Okay, ma'am. Yes. And madam, for to confirm this wound healing, which type of assays you carry out in vitro and in vivo? I'm sorry, I'm taking your time. Just 2 minutes.
You'll have to read my papers. We've just published it. It's too long an answer to go into.
Yes.
There are many potency assays that we do.
Okay.
So we do in vitro as well as in vivo. It doesn't stop there. It's a continuous evolution. We did, you can choose the model that you want. So we did excision. Now we are going to go into a burn model of wound healing. So there is a an ulcer model of wound healing. So it depends on what is your final application that you want to develop, and you have to first prove it in an animal model, then do the safety, and then you progress to human clinical trials.
Thank you, madam. Thanks a lot. Excellent talk, sir. Great talk. Thank you so much.
Caroline,
Yes.
really a great journey of yours, I should say. Right from the lab, you were able to take your product to the market. So I have just few questions for you, actually, with regard to this BIRAC grant. Once we get the grant, is it necessary, for example, this is a question from all faculty members, especially point of view, that we are employed with DPU, suppose we say, and we get a BIRAC grant, then what is the terms of these BIRAC grant? Can we continue our position at the institute and parallelly incubate with any center which is registered under the BIRAC?
I think ideally you should be ready to give up your faculty position and set up your company within 18 months.
Okay.
You would also sometimes what happens is there should be no conflict, because sometimes the institute feels that this IP belongs to the institute. So you will need to have an agreement with your institute about how this IP is going to be shared and commercialized. If once you reach commercialization, is there a profit sharing or a royalty that comes to the institute or something like that? Or it need not be. I mean, it depends on how good is the relationship and understanding. Also, there are, like, for example, IIT. IIT has its own in-house incubation sign. So, or NCL in Pune, they have their own incubation center. So I don't know if you have an incubation. I know that you mentioned there is an incubation cell, but do you have a facility for incubation?
We have registered our IIC cell with the MHRD and also with SPPU, but formally we have not started any incubation at our institute. So
okay, so I don't know whether there is a conflict of doing it in the same institute or it has to be in another institute. I have not really gone into it. That probably, if you write to NCL or Venture Center, they'll be happy to share these details with you. If you need a contact person also, I can share an email where they are happy to answer such questions. Yeah. That's easily done. So if you know, then you're mentally prepared that yes, I have to leave my job and I have to start it. But still, it's a lesser risk because you're not leaving your job till you get the grant, right? So you know that, okay, part of the journey is still funded. That still gives you months to figure out the next round of funding when you're going to hit the market. For us, the gestation period was really long because it's a biotech product, and anywhere between it takes 10 to 15 years to hit the market. So in our case, we always thought that instead of being dependent on external grants, it would be better to be self-sustaining. And that was the reason why we went after the stem cell cosmetics. We said this is a low-hanging fruit which we can commercialize and get to market very fast. So this is the sad part of the story, where we were innovating so fast and so quick that we were ahead of the regulatory curve. So till today, I mean, this this dialogue with CDSCO has been going on from 2016. I actually sat on the stem cell drafting committee to, section 13 is mine, where we said that stem cell proteins can be used for wound healing and also for cosmetic applications. They included it in the stem cell guidelines. In 2019, Bureau of Indian Standards included this ingredient as an ingredient for cosmetic products. But till today, I have not got the approval, domestic approval, to sell the stem cell based cosmetic. But I have got the export license to do it. Now it's a catch, because in your own country, if you are unable to sell it, I mean, it's difficult to sell abroad because you have to have a free sale certificate. They'll ask you, is it sold in the country of origin? So there are a lot of regulatory hurdles that I would say. This is another part of research that I would advise you to do. Apart from your market survey and your market research, also see from the regulatory. I think from your side, it should be more simple because it's chemistry, it's a molecule. And CITESCO and FDA and all, they are used to dealing with chemical entities. They are not used to understanding stem cells and live cells and proteins. So our journey becomes tougher because first we are educating them and then actually guiding them as to how to give us approvals. You know? So that becomes a sort of painful part of the journey.
Probably in case of your product, because you have taken your product for a cosmetic application, you may have not had to do a formal clinical study, or you had to prove it through clinical experimentation.
We didn't have to do a clinical study, but we did it. Those were the results that I showed you. So we actually did it to prove the safety and efficacy. It was a small sample size, but since it was something which had not been done before, as a company, we felt that we need to really have the proof of the pudding, you know, to show that this is something that works. Because otherwise what happens is, and I always caution people, when you're talking about stem cells, it's not a magic pill. It's not like a one-stop shop. You have to develop that application, that specific application. So if you talk about stem cell biologists, each one would have his pet project. So somebody might be working on diabetes, or like people like me would be working on skin related problems. So each one becomes a different application.
So now in case you want to expand your product to the therapeutic application, like actually for wound healing, probably I think you will have to again prove it.
That is a straightforward route, because that goes through, you know, animal trials, clinical trials. So
yes,
that is okay.
Okay. And other than BIRAC, are there any other agencies that support in such ventures?
No. So now all the others have come under the umbrella of BIRAC. It used to be earlier, you know, DST, DBT and all that. It's all been combined under the umbrella of BIRAC.
Okay.
So if you go to the BIRAC website, you will get a lot of information about the call for proposals. You can see the previous calls for proposals and also the kind of studies that have, there's a compendium, BIRAC compendium. So you can actually go and see what kind of studies have been supported. You'll get an idea of, you know, the strength of the proposals. So you would be able to evaluate yourself whether, yes, this is going to work, or does it have very important, two important things when you write the BIRAC proposal is IP protection and commercialization protection. These two things. So BIRAC wants you to take some money and then, you know, start running. Yeah, that's what they want you to do.